Ion exchange chromatography separates molecules based on their overall charge at a given pH. The resin inside the column carries charged groups, either positive or negative, and proteins with the opposite charge will stick to it more strongly. When you load a protein mixture onto the column, some proteins bind tightly, some bind weakly, and some don't bind at all, depending on their charge. To get everything off the column in a controlled way, the salt concentration or the pH is slowly changed. As this happens, the ionic interactions weaken and proteins elute in order of how strongly they were held. Because even small differences in charge can change how long a protein stays on the column, IEC is great for separating variants that look almost identical by other methods, like different glycoforms or slightly modified proteins. Upon researching this method of chromatography, I came across an incredibly significant discovery made through ion exchange. When researchers passed a sample of hemoglobin through a cation exchange column in 1958, they noticed a small fraction of the sample eluting later than the rest. This was HbA1c, a glycated form of hemoglobin with glucose attached to the N-terminal valine of the B chain, slightly altering the molecule's charge. This discovery led to another very important one. In 1968, it was found that HbA1c levels are elevated in diabetes patients. It could now be quantified and used reliably as a biomarker to diagnose diabetes. It is now considered indicative of diabetes if an individual has greater than 48 millimoles per mole or 6.5% of glycated hemoglobin. Aside from this accidental discovery, we have also used and continue to use IEC in large-scale manufacturing and regulatory processes in labs. For example, it is used as a standard purification step for monoclonal antibodies. These are commercial, laboratory-made antibodies which are all identical and made to recognize one specific antigen. Monoclonal antibodies work in a few ways. They can block signaling pathways, neutralize inflammatory molecules, mark cells for destruction by the immune system, and deliver drugs to diseased cells. They are usually made by fusion of a B cell with an immortal cell line or through recombinant DNA methods. Ion exchange chromatography is used after protein A capture, which I'll talk about more in the next section, and it removes aggregates, charge variants, host cell proteins, and DNA. Monoclonal antibodies are widely used to treat cancer, autoimmune diseases, infectious diseases, cardiovascular diseases, and neurological diseases. In an oncological example, the monoclonal antibody trastuzumab binds to the HER2 receptor on cancer cells, as some breast cancers overexpress HER2, which causes uncontrolled cell growth, and both blocks growth signals and marks the cells for immune destruction. An example of an autoimmune disease treated by monoclonal antibodies is rheumatoid arthritis, which destroys joints with inflammation caused by the cytokine TNF-alpha. The monoclonal antibodies adalimumab and infliximab bind this cytokine and neutralize it. Infectious diseases such as COVID-19, Ebola, and RSV have been treated with monoclonal antibodies as well. They mostly prevent viruses from entering cells in the first place. For example, the MAB114 that binds to the Ebola glycoprotein. The last application I want to mention is for migraines, which are triggered by the release of calcitonin gene-related peptide, causing pain signaling and blood vessel dilation. CGRP inhibitors are used to block this from happening. In addition to purifying monoclonal antibodies, ion exchange chromatography is used to manufacture recombinant insulin, which helps treat diabetes patients who may have been diagnosed in the first place thanks to IEC. Enzyme replacement therapies are also made using IEC. These are commonly used to treat lysosomal storage disorders, wherein a single defective enzyme could cause a buildup of toxic substrates in cells. Patients are administered these lab-produced enzymes intravenously, and the enzymes are taken up by cells, often by mannose-6-phosphate receptors, and are transported to lysosomes to degrade the toxic substrates that have accumulated. To get more specific, one of the enzyme deficiency diseases that affect humans is Gaucher disease, which is a deficiency of beta-glucocerebrosidase that leads to an accumulation of glucocerebroside, ultimately causing anemia and skeletal issues. Another disease is Pompe disease, a deficiency of acid alpha-glucosidase, causing glycogen buildup in muscle, leading to cardiomyopathy, or progressive muscle weakness. In conclusion, ion exchange chromatography was a critical invention that led to a cascade of new treatments alleviating the pain of a wide array of diseases.